Good afternoon, everybody. Do we have with us the speakers, Dr. Konstanti and Dr. Gravanis? There, okay, thank you very much. My name is Pavlos Neofidou. I am the chairman of the Health Committee of the Cyprus Biological Society, which sponsors this session of the Congress. Our first lecture will be given by Professor Dr. Maria Konstanti. The title of her talk is The Neuroprotective Effects of Olea europaea, Crocus sativus, and Salvia species. Dr. Konstanti, thank you.
I would like to thank first Professor Patrikios and Elizabeth Johnson and all the members of the organizing committee for the invitation. I'm honored and thank you. And it's a great pleasure for me to participate for one more year in this wonderful conference. Well, let's see. Okay, this way, okay. Here. It's over 50 million people around the world that live with dementia. And in this number are not included the patients at prodromal or even preclinical stages. And the goal of treatment for these patients is to stop the disease progression and prolong the period of full autonomy in daily life activities. Unfortunately, there is no effective cure so far, despite the intensive research. The promising molecules for the development of new drugs unfortunately fail in clinical trials. In Alzheimer's disease, there are several neuropathological features, including the accumulation of beta amyloid proteins that form plaques within neurons, the hyperphosphorylation of tau proteins that form these neurofibrillary tangles, mitochondrial dysfunction, calcium deregulation, blood-brain barrier leakage and neuroinflammation, and all these conditions lead to the neuronal degeneration and death, mainly of cholinergic neurons in the basal forebrain and the neocortex. These neuropathological conditions lead in the progressive impairment of behavioral and cognitive functions, and finally to dementia. There is intensive research also on herbs, on the application of herbs, and since ancient years started, and including salvia, but nothing came so far. In our study, we investigated the potential neuroprotective effects of Olea europaea, Crocus sativus, and different species of salvia. This study, a preclinical study, employed various experimental models of the disease, and took place in collaboration with the Institute of Biosciences, the University Research Center of Ioannina, the Department of Pharmacology of the University of Crete Medical School, the Institute of Molecular Biology and Biotechnology of the Foundation of Research and Technology of Crete, the Department of Pharmacy of National and Kapodistrian University of Athens, and Demokritos. The in vitro study we conducted employed the human SH neuroblastoma cells that were differentiated with retinoic acid to cholinergic neurons. And these differentiated cells pretreated with the extracts of the plants or the active compounds for 2 hours before being exposed to the to the amyloid or superoxide. And the viability of the cells was assessed with the MTS assay. The results of the in vitro study indicated that the extracts of Crocus sativus, the active compounds tested, had no toxicity. Oleuropein and the extract of olive leaves increased the viability, cell viability, by the total phenolic fragment of the extract L-ocanthal displayed very significant toxicity. The salvia, different species extracts, had no toxicity at low concentrations, but we've seen toxicity at the higher concentration. When these neurons, the differentiated cells to cholinergic neurons, when these neurons were exposed to superoxide, to oxidative stress, their viability reduced to the half, almost to the half, and exacerbated by the extracts of Crocus sativus and the active compounds. The total phenolic fragment of the extract of olive leaves, and the oleucanthal, but oleuropein, the most important active compound of Oleuropea, protected the cells. The salvia fruticosa and officinalis extracts also protected the cells, but all the other salvia extracts exacerbated this toxicity. When the cells were exposed to beta amyloids, we've seen again toxicity, reduction of the viability, more than to the half, and the transcroquin 3 and 4, active compounds of Crocus sativus, protected the cells. Oleuropein again protected the cells from this toxicity, beta-amyloid toxicity, and the phenolic fragment and the oleucanthal exacerbated strongly the toxicity. From the salvia species, the salvia argentea and verbenaga extracts protected the cells, but all the other extracts exacerbated the toxicity. In order to see if there is any direct effect on the aggregation of amyloid proteins in these cells, we ran the theoflavine assay, and we observed that the transcroquin 3 and 4, oleuropein, the extracts of salvia argentea and Crocus sativus, several diterpenes, triterpenes, methoxyflavones, phenolic compounds, the rosmarinic acid scutallerine, some of the active compounds of salvia protected the cells against the aggregation of beta amyloids in the culture containing these neurons. In the in vivo experiment, we used the 5X1 transgenic mice that bear 5 mutations in corresponding genes that are very important for the pathology of the disease. And these transgenic mice develop rapidly severe amyloid pathology, in particular accumulation of alpha-beta 42, the most important type of beta amyloids, starting from the age of 1.5 months, and at the age of 4 to 5 months, these transgenic mice develop a phenotype approaching that of the disease, of Alzheimer's disease. The transgenic mice at the age of 2.5 to 3 months received the extracts of the medicinal plants I mentioned before in a jelly for 33 days. And at the end of this period, the mice were submitted to various behavioral tests in the open field, novel object recognition test, novel object location test, in order to assess if there is any effect of the extracts to basic cognitive functions. And we observed that the extracts reduced the distance traveled by mice in the open field, the ambulatory and vertical counts, except the crocus, and they increased significantly the discrimination index in the novel object location and recognition tests. The in vitro data indicated that the salvia different species, and Crocus sativus and Olea europea, contain several compounds that display significant but dose-dependent antioxidant and neuroprotective properties, and in particular, oleuropein, transchrokin 3 and 4, scutellarin, rosmarinic acid, and several diterpenes and triterpenes. But we observed that there is a diversification of the antioxidant and neuroprotective effects of the distinct salvia species. And I have no doubt that there is this diversification in the other plants, medicinal plant species we tested, but we didn't have these samples to test. In the in vivo experiment, we observed that the salvia, Crocus sativus and Olea europea extracts improve the spatial and recognition memory of the transgenic mice. Taken together, this data, the current data, and the data from previous studies indicate that the medicinal plants we tested can mitigate the oxidative stress and alpha-beta-induced neurotoxicity, and potentially are promising to be given as complementary to the traditional treatment. And I think that it's more significant, it's more important to say that for the development of new drugs, because they contain many active compounds with great potential. We should bear in mind, though, that all the medicinal plants, and this we tested, contain also toxic compounds that can significantly reduce neural viability and exacerbate the oxidative stress and alpha-beta-induced neurotoxicity, and therefore they should be used with caution and in moderation in herbal preparations. In addition, we should bear in mind that the compounds they contain may also interact with the prescribed drugs, and this is of paramount significance in particular in the elderly and patients on medication, because these active compounds can modify the efficacy of pharmacotherapy and potentially induce toxicity. The data I presented here are part of the doctoral dissertation of Aristides Kofinas, and for the conduction of the study, Professors George Leoderitis and Katerina Antoniou had a very substantial role. Christina Andriopoulou and Katerina Palianga also participated in this study. Thanks a lot.
Thank you, Professor Constantin. Is there a question from the audience? No questions? Okay, thank you, Professor Konstanti. My co-chair, Dr. Tsvetanova, will introduce the next speaker.
Hello, welcome to our session. It's my great pleasure to introduce the person that's not paying attention to me right now, Dr. Achilleas Gravanis. He's a professor at the University of Crete Medical School. He's a professor that visits us quite often and teaches our students as well. He goes and teaches in Boston, so he is teaching pretty much everywhere. But more importantly, Dr. Gravanis is also working on some very exciting discoveries. He has talked to us in the past about his work on spinal cord injury, and today he's going to talk to us about the combination of TrkA, TrkB neurotrophin receptor activators in Alzheimer's disease.
Thank you, Eva, for this kind introduction, and I want to thank also my friend Yanis, who keeps me posted and present in this exciting, actually, event for, Yanis, 12 years. We are growing together, actually. So, my colleague, Professor Konstanti, made my life very easy because she introduced you to this devastating disease, which will be the disease of the future, and is a result of our success, of the success of medicine for the last 20 years. So I will only underline the three biological endpoints of the disease. The patients lose various types of neurons, and so we observe neurodegeneration. The patients have a very important neuroinflammatory component of their disease, which was shown actually for almost all neurodegenerative diseases the last 10 years. And these patients also have problems with their myelin. They have lots of demyelination, a new event that was shown the last actually 3 to 4 years. It is frustrating because in almost all neurodegenerative diseases, we don't know the etiology, the cause of these diseases. So for us, pharmacologists, we cannot design actually targeted therapies. So the only option we have so far is to try to help our patients with symptomatic treatments. And this is exactly which is our focus in our group in Crete, and our spin-off company, By Nature Ltd, which owns all the IP and the compounds I'm going to present today. So we design molecules, multidynamic molecules with anti-apoptotic neuroprotective activity, and at the same time these molecules can block the activation of microglia and induce phenomena that can block in situ neuroinflammation. And lately, these compounds were shown to be very effective in inducing neurogenesis in vitro, ex vivo, and in cell cultures. One of the most important actually mechanisms by which our brain controls the survival of neurons and microglia cells is through neurotrophins. Neurotrophins are polypeptidic actually agents produced in our brain and the peripheral nervous system. We have NGF, the first described neurotrophin by Levi Montalcini and Professor Cohen, and they got the Nobel Prize, actually when I was born. These two neurotrophins are recognized with specific receptors, membrane receptors, TrkA for NGF and TrkB for BDNF. They are involved in almost all important actually pathways in our brain, and they are very important for sustaining neuronal survival for all ages of a human being. If we lose the capacity in the brain to produce this type of agents, we are going to start degenerating in our brains, and they are shown to be at low levels to almost all neurodegenerative diseases. So for us pharmacologists is a good target for a symptomatic actually use of this knowledge. I'll go through the already published results involving neurotrophins in Alzheimer's disease. NGF promotes the survival of basal forebrain cholinergic neurons, very important cell population affected by the disease. And of course, in AD, the levels of NGF, as I mentioned already, are heavily decreased. NGF was given intranasally in animal models of the disease, and they were found very effective in blocking some of the symptoms of the disease in these animal models. And also there is a formulation of NGF gold nanoparticles which was effective in reversing some of the damages of these animals. The levels of BDNF, as is the case for NGF, was also reduced in the AD brain, including humans. And in animal models, if you increase the signaling of BDNF, you can reverse some of the losses in cognitive capacities of these AD animal models. And transplantation of stem cells into the brain of triple transgenic AD mice can equally reverse some of the tau pathologies related to the disease. So our group, in collaboration with my friend for the last 20 years, Theodora Kalogeropoulou, an organic chemist at the National Research Foundation in Athens, designed and synthesized a big library, chemical library of small molecules, highly lipophilic to pass the blood-brain barrier, mimicking some of the effects of NGF and BDNF. And I'm going to present the data actually which were published this year related to AD. Cyprus Medical prepared his PhD thesis in Crete, studying the first of our compound, BNN27. Cyprus now is in Boston. He spent some time in Harvard, and now he's in a spin-off company developing new therapies for AD using specifically biotechnologically designed monoclonal antibodies. So this molecule can mimic NGF in activating TRAC receptor, phosphorylation of the receptor, and internalization of the receptor and activation of specific gene networks. And the next step was to use a very useful, actually, animal model of AD. Maria described some of the properties of this molecule. It's a 5-transgenic molecule possessing 5 mutations related to human AD on APP and beta secretases. And it develops a specific phenotype. It does not, it simply simulates the disease because Alzheimer's disease is not a single disease, it's a panel of diseases with some common aspects in terms of neurobiology, but we have different types actually of Alzheimer's, and we are now starting by studying clinical data to realize that the disease is not only one actually. And that's why it deserves different treatments for each one of it. So these animals accumulate beta amyloid. You can see strong astrogliosis in 2 months, synaptic loss, neuronal loss in the hippocampus, and of course deficits in their recognition, and LTP function in 6 months. We gave the BNN27, the formulation was a pellet, and the pellet was given for 60 days, and then we were following the animals behaviorally, and we tested the effect of the compound in vivo given in working memory, spontaneous alteration performances, and the tests that Maria described. The work will come out in molecular psychiatry next week, so for those who will want to read in details the paper, they can go there. So the compound was very effective in reversing almost 100% of these effects. We were very surprised because in the literature there were many compounds that they were used to reverse these phenotypic behavioral changes in this animal model, and none of them were so effective in reversing this phenomenology. BNN27 decreased the formation of alpha-beta plaques in the hippocampus of these animals and increased the synaptogenesis and decreased astrogliosis, two components, pathological components actually of the disease. Interestingly enough, BNN27 promoted neurogenesis in the hippocampus, specifically in the data gyrus of hippocampus, and this induction was very, very efficient. The compound protected mouse hippocampal neurostem cells in culture. We wanted to mechanistically study the effects we have seen in vivo by using cell cultures of neurostem cells, hippocampal neurostem cells, sterilely differentiated, coming from adult animals actually. And the compound was shown very effective in protecting these cell cultures against beta-amyloid toxicity. It reversed also beta-amyloid toxicity in neurons coming from embryonic tissues, from hippocampal cultures, and was shown to have a very protective effect, which was blocked almost 100% by a pan-TrkA receptor. I remind you that TrkA receptor is the receptor that binds NGF and of course our compound. We have done comparative proteomics analysis in wild type versus 5XFAD animals, untreated and treated with the compound, and we surprisingly and very indexed shown that the compound can mimic actually the effects of NGF. Not only that, it affects many, 35 out of 80 genes related proteins, related to the pathology. The next step was to study the BDNF neurotrophin. So we have shown, and it was published last month in GLIA, that BDNF is very important in protecting the brain of these animals against demyelination. And we have specifically shown that demyelination is decreasing through the lifespan of these animals. Ioanna Zota, one of my PhD thesis, did a very tedious work on that. We went again in vitro to find the mechanism by which this is happening. So we cultured oligodendrocyte precursor cells. Let me remind you that OPCs give rise to mature oligodendrocytes which produce myelin. So beta amyloid was shown and it was published many years ago to be very toxic for these cultures, and BDNF could reverse effectively enough this toxicity. This phenomenon was also reproduced in primary cultures of mature oligodendrocyte precursor cells in wild-type animals. Having this data, we again worked with Theodora Kalogeropoulou, and we designed and synthesized a large chemical library of BDNF mimetics, TrkB receptor agonists, and one of them is very effective in inducing proliferation of human cortical neuroprecursor cells in cultures. It is very effective in decreasing proliferation and survival in mouse adult hippocampal neurostem cells in culture, and also increase the proliferation and survival of mouse embryonic cells in culture. We have done also a comparative oncology of the effect of this compound compared to BDNF to find out if it has the capacity of mimicking some of the effects of BDNF on gene expression, and indeed we had very interesting results. The compound can mimic BDNF in controlling actually proteins related to APP. It can regulate the genes regulating the sensitivity of this tissue to endogenous BDNF, in other words TrkB, and also regulates many of the proteins known to be involved in the disease. With collaborations in Europe and in the US, we have expanded our chemical library. Now we have many synthetic compounds mimicking the actions of BDNF and as agonists and activators TrkB receptor. So to conclude, our idea is to design and test a combinatorial treatment for Alzheimer's, symptomatic treatment, focusing on diminishing neuronal apoptosis, demyelination and activation of oligodendrocytes, neuroinflammation by blocking glia cells, and of course, in an effort to reverse some of the damages by inducing in situ neurogenesis to replace the damaged neurons with new, fresh, normal ones. And these are the collaborators and students that perform all this work and our family. Thank you very much for listening.
Are there any questions? Yeah.
Thank you. Yes, for this wonderful lecture, and I must say that I'm impressed how many disciplinary practice was surprisingly positively with what the answers that are uncovered. And my question would have to do with amyloid, beta amyloid. Is it, do you know already, have you established whether it acts on amyloid or indirectly to the mechanism results in the reduction of the amyloid plaques?
Vangelis, let me make a statement which I think is useful for an audience that is not very informed on the disease. Beta amyloid was considered for many years as the pathophysiology and the etiology of the disease, which is now not the case. Actually, we know now, even Robakis, Professor Robakis, who was one of the first proposing beta amyloid pathology for AD, is convinced enough with his data that is an epiphenomenon of the disease. So something else is happening, or many other things are happening through our lifespan, and then you have this toxicity. So in our case, there are data in the literature showing that neurotrophins can affect the process of APP gene and also the post-translational processing of the end product, inducing also secretases. So most probably, we didn't test it, but most probably this is the mechanism by which these compounds work through the regulation of the expression of APP gene and the post-translational processing through gamma secretases.
Minor correction, John Hardy was the first person to the amyloid hypothesis. But more appropriately, I agree with you about the fact that it is a tombstone of cell death. But now tell me why your molecules aren't tombstones, why the decrease in activity of these agents isn't simply a result of fewer cells that are alive. And therefore is also a tombstone effect. Why do you think it's a primary event which causes the disease?
You're right. I mean, because we don't know the etiology, the only thing we know so far is that two mutations in the ApoE gene are heavily involved in linkage. Actually, they are linked to the disease. So
that's a correlation, though. That's a correlation.
That's a correlation, absolutely. Yes. So we have no idea on the pathophysiology of the disease in terms of the etiology. So that's why I mentioned at the beginning, until we find the etiology, we should focus out of the box thinking on symptomatic pharmacology, actually. So, and because it's a very complicated disease with different types of clinical manifestations, we have to be multifaceted. We have to target different aspects and biological points of the disease to increase the possibility of controlling the disease. That was our hypothesis, and that's why we reasoned this way. Symptomatic treatment, multifaceted symptomatic treatment.
But that may just be a function of think to label all those as Alzheimer's disease could confuse people. I think that I agree with you exactly that there are many causes of dementia, but I think there's likely to be one cause of Alzheimer's disease as described by the neuropathologists, particularly at the level of the neural plaques. It's a great area to be in, and I very largely agree with you, but I wouldn't call all dementias Alzheimer's disease. Would you call the dementia you get like Cassius Clay with his punching, plus a bit of Parkinsonism associated, would you call that Alzheimer's disease? Or would you call that dementia?
Yeah, I agree with you. And the more we get in terms of clinical data and linkage analysis with different gene expression profiles, the more we realize that we have different types of the disease. So we have to focus on each one of it. And that will be, I think, the future, and it will be very helpful for designing a new therapy for each one of them.
Thank you.
It's exactly the same story with Parkinson's disease. Sorry. It's exactly, you've mentioned actually that. Yeah.
There are a few more questions in the audience, but we are running out of time. So please, Dr. Gravanis is always willing to talk about research and not only. So meet him in the coffee breaks and talk to him about it. Thank you very much.
Thank you.
We now invite Dr. Marius Panzaris and Dr. Tassos Karandreas to chair the following segment.
So, good afternoon. I'm Tassos Karandreas. I'm a professor in surgery, general surgery, and special thanks to the Deputy Dean Ioannis Patrikios for his kind invitation. So, the next session is concerning neurology, neuroimmunology, neuropharmacology, and chronic diseases. The lecturer is Professor Marios Panzaris, Senior Consultant Neurologist, Head of Neuroimmunology Department, Cyprus Institute of Neurology and Genetics. Please, Professor, you may proceed.
Thank you. Let's wait for the presentation from here. Thank you. Thank you, Patrikios, for the invitation. Okay, thank you. Now, I'm going to talk to you about the stem cell treatment from the bone marrow concerning multiple sclerosis patients. And this is an unmet need that we have since we have a lot of patients that they do not respond to any treatment that we have until today. So the reason that I have decided to do this presentation is because we are now starting in collaboration with the German Oncology Center to establish a protocol and give this treatment in Cyprus. So, as most of you know, the bone marrow is the lining within the bones, especially in the vertebra and the ribs and the pelvic bones and the sternum, and it consists from two different types of marrow, the red one and the yellow one. From the red one, red blood cells, platelets, and white blood cells will originate, and from the yellow marrow, the stromal cells and the mesenchymatic cells will originate. Now, from the red marrow and the hemolytic stem cells and the amyloid and the lymphoid progenitor cells of the cells of the blood will originate. And the important thing from the mesenchymatic or stromal cells is that these cells can give rise to muscle, cardiac muscles, neurons, and other cells if you treatment in the lab. Now, what is autologous hematopoietic stem cell transplantation? It's a procedure that has several steps. In the first step, we mobilize hematopoietic stem cells from the bone marrow, giving the patients intravenous cyclophosphamide and subcutaneous colony-stimulating factors. And after several days, we proceed with collecting the stimulated stem cells through a procedure called leukapheresis, and we keep them freezed as a cryopreservation method for the next step, where several days later we condition the patient with several cytotoxic drugs in an effort to kill the abnormal immune cells and even the bone marrow cells. And on the later stage, we give back the hematopoietic stem cells that we collected, and within several weeks, up to 4 weeks, the patient's immune system is reconstituted back, and the patient can go home and follow for several period for any side effects from the treatment. Now, the conditioning is the treatment that we give in order to kill the abnormal immune cells. Usually today we use the intermediate intensity protocol, where we only give cyclophosphamide and thymokyne, antithymokyne globulin. And through this method, we only kill the periphery abnormal cell and we not destroy the bone marrow. This is called the intermediate method. Now, what is happening after we give back the hematopoietic stem cells? In several months, we see that T cells are starting appearing in the periphery, and later under the action of thymus gland, more T cells and B cells are appearing in the bone marrow, and even later we can see that there are some normalization of gene expression leading to the restoration of immune tolerance, where the abnormal immune cells that were not recognizing our own protein as our own protein, they are now functioning very well without causing any neuroinflammation. So what is the aim of the autologous hematopoietic stem cell transplantation, SMS? Is to destroy the abnormal immune system and restore it with normal functioning cells. And in contrast to the other treatments that we do have, and they are very effective, is given once off or is done once off, and it lasts for up to date, we have 10 years of effectiveness. What is doing the autologous hematopoietic stem cell is that it has a very rapid effect with rapid reduction of relapses, activity of the disease, and the progression of the disease. The first study was done in Thessaloniki in Greece, and it used to be done in patients that they had progressive disease with very severe disability. There were 15 patients, and they get a very strong conditioning with a lot of immune suppression. And you can see that within the first year, most of the patients presented a dramatic improvement of their disability. As the years went by and we get a lot of information about the effect of this autologous hematopoietic stem cell transplantation, we changed our opinion, and from the progressive patients that we started giving the drug, we now give the drug in the relapsing patients, the patients that they have frequent attacks of the disease, the relapsing type, and in the small percentage of the patients that we give with the progression of the disease, these patients, they do also have activity with relapses or new MRI evidence when you do the MRI scan. And recently, we have finalized our method to the non-myeloablative protocols using only cyclophosphamide and anti-thymocyte globulin. So we only destroy the peripheral immune cells, not the bone marrow, prodromal or stem cells. So today, in our meta-analysis, we know that autologous hematopoietic stem cell transplantation can reduce the frequency of relapse activity up to 70 to 100%. It can reduce the progression of the disease up to 100% for 5 to 10 years with no major side effects. And as you can see, most of the most effective drugs that we have can lead up to 50% reduction of the activity of the disease, whereas the autologous hematopoietic stem cell transplantation can lead up to 80 to 93% reduction of disease activity. So it's very effective. We have also seen that patients with relapses, that they also have a progressive nature of the disease. They produce a significantly less progression of the disease when they have the autologous hematopoietic stem cell transplantation compared to other medications that we have, and even they present some improvement of their disability. The mortality rate of the autologous hematopoietic stem cell transplantation in the beginning, in the 90s, was very high, 7.3%, but today this mortality rate is less than 0.1 to 0.2 because of the improvement of the method and also of the patient selections, because today we know that patients that have many years of the disease, very high disability rate, if they are very old in age and they have the non-relapsing MS type of the disease, they have the most chances, or they're in great danger for presenting mortality from the method. So today we have a protocol that we follow, and the method of treatment is given in the relapsing remitting MS patients, in progressive MS patients with relapses or MRI activity. The patients have to be young, the short disease of the duration, less than 10 years, and they have to give a consent of their treatment, and also they need to have no other comorbidities that would put them in danger for increased mortality. So today, most of the international MS societies and neurologists, they advise that we can give this method of treatment, the autologous hematopoietic stem cell transplantation, to patients with active relapsing MS patients or relapsing progressive MS patients that they do not respond to any of the effective treatments that we have. And these patients must be young, less than 45 to 50 years old, and also they need to have the duration of the disease no more than 10 years. Another advice that we do have from the International Society of Bone Marrow Transplantation is that we need to have specialists in hematology that they know how to do the procedure. We need to have neurologists that they will have experience in the follow-up of these patients. We need to have a single database and put this data on for long-term observation. And also, we need in the future to have more and more ideal procedures, either in mobilization of cells or in the conditioning of patients. And finally, over the last few years, we do have comparative studies comparing the autologous hematopoietic stem cell transplantation with the most highest effective treatments we have from the beginning to see which is the best effect, which is the best treatment in the long term of patients with MS, because a lot of neurologists, including me, we believe that we need to fight the disease drastically from the beginning. Now, what about these mesenchymatic stem cells that we do have in the bone marrow? A lot of studies have shown that these cells have a lot of secretory capacities. They do exert a very fantastic and dramatic paracrine function where they are going, and they secrete neurotrophic factors, they secrete cytokines, chemokines, even small particles of that as extracellular vesicles are moving around in the area where they are going, and they exert actions like remyelination. They modulate the cells of the adaptive immune systems, the T and the B cells. They regulate the innate immunity, leading the microglia to a more pro-inflammatory, anti-inflammatory state, so it's improving neurogenesis. They have a fantastic migration capacity to go through the blood into the areas of the brain and travel to the areas of the inflammation. And finally, we've seen that they reduce the toxicity of iron-loaded within the cells in the brain that are found around the MS lesions. As you can see in this case, you can see that you need to give repeated infusions of mesenchymatic stem cells because their action is transient. And as you can see, while you are giving the infusions, the disease of the activity remains low, but unfortunately, if you stop it, you see that the disease is recurring. So you need to give it continuously. All meta-analyses in studying patients with progressive MS have shown that the disease is effective in these patients with progressive MS, either stabilizing or improving the patients, but this analysis includes also patients with progression and disease activity. If you narrow down the meta-analysis and you only include patients with progressive MS without any evidence of activity, meaning no relapses or no MRI findings of new lesions in the brain, then we see that there are no significant statistical evidence of any help of these mesenchymatic stem cells in this type of patients, which is the type of the patients that we need to find a treatment because we do not have. And why is this happening? We believe that MS and all the models that we use in experimental allergic encephalomyelitis are not the same. MS is only in humans. The immune system of humans is completely different from any animal. The second one is that mesenchymatic stem cells can be collected from bone marrow, can be collected from adipose tissue, and these tissue cells present different characteristics. The third one is that these mesenchymatic stem cells that are coming from MS patients are not so good as from healthy controls. And finally, the cryopreservation of the cells leads to a less functional capacity of these cells. So we need fresh mesenchymatic cells. And the third is that all the experiments have been done in animals where you give 1 or 2 million of these mesenchymatic stem cells in a mouse of 20 grams. And if you are going into the human with 70 kilos, you need 3 billion. And all studies in humans have been done with 1 to 2 million per kilogram. So probably we give small numbers of mesenchymatic cells. And lately, we have discovered that giving IV intravenous mesenchymatic stem cells, they are trapped. The majority are trapped in the lungs. They are destroyed, and the minority go to the brain. So we thought of giving them intrathecally. So we do lumbar punctions, and we give the mesenchymatic stem cells intrathecal, and they go very well into the brain, where they produce their secretome, the cytokines, the neural growth factors, and other substances where they exert their neurogenesis, remyelination, and so on. But this is a transient effect, so you need to give repeated intrathecal infusions of mesenchymatic stem cells, and unfortunately, this leads to inflammation of the arachnoid. So it's causing arachnoiditis, and this is a very severe complication. Lately, we have started thinking of using not the cells, but the secretome, the substances that the mesenchymatic stem cells are producing, and this is done very easily through the intranasal route, because whatever you give from that route, it can go directly into the brain, and perhaps this might be the solution in the repeated infusions. So thank you very much for listening.
Thank you very much, Professor. Any questions from the audience, please? So, thank you very much. We may proceed to the next lecture. It's about systems biology dissection of stress-related mental disorders across brain regions, omic layers and cell types by Professor Nikolaos Daskalakis, who is Associate Professor of Psychiatry, Harvard Medical School. Hello, Professor. I don't know if you listen us. We cannot listen to you. I think we have a dysfunction. Can you solve this problem, please? Is that problem from us or from Professor?
Can you hear me now?
Yes. Yes, we can hear you.
You can hear me now?
Thank you very much. Hello, Professor.
Good morning, Professor. Let me share my slides. Can you hear can you see the slides in full screen? Can you see the slides?
Yes, we can see them. It's okay.
So I will be talking to you today about, I see myself with an echo. So, in stress-related disorder, I'm working on stress-related disorders, and it's important to understand that a way of understanding stress-related disorder is to remember ourselves about the stress response. All mammals, including humans, we have a hardwired response to stress. So when we are exposed to a threat, here is a photo of a tiger. Behaviorally, we have to respond very fast with a fight, flight, or freeze response. And that has to happen very fast because we have to have a behavioral response to what is life-threatening. This is mediated from the autonomic system, and it's very fast. And then something that is also very fast is when the threat is gone, is that we also have to adapt and return to homeostasis with systems like the HP axis and the end product cortisol that is mediating the return to homeostasis. Something happens in stress-related disorders, something around individual variation, that individuals with stress-related disorders stay in that state of activation, although there is no more stress in their environment. A way of understanding individual variation is through GWAS, genome-wide association studies. And these genome-wide association studies are trying to connect the genotype with they're trying to connect the genotype. I was trying to create. Okay, so they're trying to connect the genotype, which is the millions and millions of letters of the DNA, with the phenotype, that is the binary variable, having or not having the disease. Statistically speaking, this is very hard, and because it's very hard, you need a lot of power. And here is in the middle, you can see approximately what is the number of cases you need to have to identify the so-called significant loci for disease. PTSD, which is the disorder that I study, somewhere in the middle of schizophrenia and depression. Schizophrenia, after having 17,000 cases, now it has approximately 300 loci. MDD, which is much more environmentally regulated, did not give so much back in the investment of the GWAS. And PTSD is somewhere in the middle, where we have identified now 100 loci. But when you are thinking about it, in order to discover treatments, understanding disease better, you need to, at the same time, understand the biology of disease. And this you can do with a system biology approach that doesn't go directly from genotypes to the phenotype, but also introduces intermediate steps. For example, it introduces the relationship of genotype to molecular phenotypes, and then how those molecular phenotypes relate to neurobiological phenotypes that you could potentially study in live humans, like neuroimaging. So bridging the gap from genotype to phenotype. One way of doing it, I will give you one example, and then you can think of like a lot of other combinations, is, for example, when you focus on gene expression, you can study the relationship of SNPs with the gene. And in order to do that, you have to measure gene expression at scale with RNA sequencing or similar approaches, and also sample the tissue of interest, in this case, the brain, and collect more and more samples. But in the end of the day, when you have these samples, you can measure the genotype, but also measure gene expression. In a simplistic way, you need to create an equation that links SNP genotype with gene expression, so a combinatorial model that can predict gene expression from genotype. And these gene expression outcomes could be as complicated as spliced RNA or linear RNA, circular RNA, cell type specific RNA, depending on the question at hand. And these models can be stored, transferred to others. And what you could then do is link genes that are in particular tissues and brain regions and cell types with disease. And this you can do it in the two ways. One way is to impute gene expression. So suddenly, when you have a biobank like the Cyprus Biobank, you can take the genotype files and convert them to impute gene expression, and then associate that with the phenotype, or going online, find summary statistics of disease, which is basically SNP associations with disease, and convert them to gene, tissue-specific gene, or cell-type-specific gene association with disease. That would make a more appealing scenario in which you can form a hypothesis, test a drug, calculate polygenic risk scores that are more biological and potentially more meaningful. This is what we did recently in a Nature Genetics paper for PTSD. So in the very up Manhattan plot, it's a Manhattan plot because it has the skyscrapers and the statistical signals go up and down, and some are big and some are small. We have did analysis to associate genes with this PTSD risk, and we start identifying particular loci that are very consistent, where we can really see mediating genes. But we are also early days, so there are particular locations in the genome that we don't have an exact understanding of what are the genes that mediate this. The obvious next step would be to actually study these gene expressions in the real tissue of PTSD and actually see what is going on. Is it the genetics? Is it the environment? Is it a combination of the two? In a recent publication in Science, we did exactly that. And this came up in May this year. And the experimental design was that we said, okay, we need to create a very big data set of postmortem multi-omic investigation of the PTSD brain. So we had 300 brains, 100 were from PTSD cases, plus minus depression. One control group that had no psychiatric phenotype, and MDD, which was a control group because MDD is the most common comorbidity of PTSD, but these cases did not have PTSD. So in these brains, we divided them into discovery and replication. We measured transcriptomics, methylomics, and proteomics from 3 brain regions. And in a subset of those, we also did a single-cell transcriptomic analysis, basically studying gene expression in the prefrontal cortex, but in every single cell of the sample. At the same time, having like biomarkers in mind, we have analyzed the UK Biobank proteomic data set of 1,500 proteins in relation to PTSD and MDD phenotypes. And at the same time, as I said, we were analyzing at the same time the GWAS of approximately 1 million people for PTSD and MDD, because in the end of the day, we wanted to see if the more critical gene expression, molecular phenotypes are different on the basis of genetic risk, or are just, or are more related to disease processes, more downstream of disease, with the hypothesis that in a disease like PTSD, you are born with some genetic background, but then you accumulate stress exposures, you eventually get the disease, eventually get the treatments. So when you die, all these gene expressions or other molecular phenotypes are influenced by all those factors at the same time. Now I want to go fast through the results. I think more important is you can access the paper and read the details, but when we compare PTSD and MDD, we want to identify which are the molecular features that are primarily different. We saw that prefrontal cortex gene expression was by far for both the most informative for this, but we also saw that in other brain regions, other molecular phenotypes had signals. For example, DNA methylation, this epigenetic regulation of gene expression in the hippocampus was relevant for PTSD, and other features were more important for MDD. Another thing we wanted to do is to actually correlate the data. One type of a correlation we did is, are the different regions, depending on the molecular phenotype, correlated with each other? So are they redundant? And the question was that there was low to moderate correlation. So the regions are not redundant with each other. They bring different aspects of biology, and also DNA methylation, that is very environmental influence, was the feature that was the least correlated between regions. Another question was, is the phenotypes overlapping? And within disease, it was important to know if die by suicide or having an experience of childhood trauma, is it producing the exact same result as the primary diagnosis analysis? And the first answer was largely yes, but depending on the particular feature you study, especially at the protein level, there were differences. Another question was, are male-specific and female-specific analysis overlapping with the primary diagnosis? And the answer was that males and females, male and female-specific analysis in PTSD largely had the same relationship with primary diagnosis, which was moderate correlation. But at the protein level, that correlation was much less. But in the MDD, we saw a sex difference there because the male analysis seems to be more in line with the primary diagnosis compared to the female analysis. And another question is PTSD and MDD overlapping as diseases? And this question is primarily driven by the comorbidity and also by the fact that genetic correlation says that the overlap, the genetic overlap of the two diseases is above 90%. So the question is, are they the same? The answer is that if you pay the price and you study the downstream biology of disease, you see that they are actually more and more different. Here at the multi-region, multi-omic level, you see they have a moderate correlation. And especially for some particular features like DNA methylation, that correlation is even low. And also, there are genes that are different between the diseases directly that are not captured by studying the primary diagnosis alone from each of these disorders. Then one other question was, can we actually correlate omic layers with each other, and how to do that? One way of doing that is to map the data into pathways and correlate them at that level, and realizing that the multi-omic signatures at the pathway level are not very much correlated. And that was a surprise to us, based on the classic biological dogma. And, going to the details to get a feeling how this can go is, we had the expectation that immune pathways would be involved in all those multi-omic layers, and that was true. But then we realized that some immune pathways could be endorsed only by the RNA levels, and then there could be other immune pathways that are endorsed from both RNA and protein, but then the direction of effect being opposite in the two. Also, other pathways that are not related to the immune system were also endorsed only by, for example, the synaptic involvement in both disorders was primarily endorsed by DNA methylation, RNA metabolism by proteins, and so forth. Giving that complex picture, in another approach to go about this, we try to kind of spatially register this data, try to understand if there was, the way you extract the tissue, you get, a sample of the tissue, but at the same time, you kind of collapse the structural anatomical layering of the cortex, and we want to see if inside the data we can capture effects of disease in different layers of the cortex. And we realized by doing this analysis that in the upper right panel that the RNA signal that had this prevailing immune dysregulation was in the very higher layer of the cortex, where you have primarily non-neuronal cell types, you have the leptomeninges and a lot of non-neuronal cell types, while the proteomic signal was in a lower, deeper layer where you have the neurons. So we formed the hypothesis that there has to be a cell type specificity happening. Another way of going to the data was to doing factorization, and we did that. And then basically we recapitulated multi-omic factors, some related to age, some not related to a particular factor, but they were different in disease. In the single cell analysis, we did primarily analysis by major cell types, although the data have seen a lot of other subtypes of cells. I have to finish, right? Did you say something? Sorry.
I'm sorry, Professor, I'm interrupting you. We are a little bit behind our scheduled program.
I will just finish in 2 slides.
Thank you.
So in the single cell analysis, we recapitulate the cell type specific signatures for both disorders. We had power to detect particular signals of the glucocorticoid receptor system, but also other signals. And we also saw that at that level, the correlation was smaller between the two diseases. And we were able to validate this with stem cells exposed to glucocorticoids. And the final thing, we had this observation in UQ Biobank that we show that the correlation of brain and blood was primarily at the RNA level of the brain with the proteins of the blood, making some hypothesis about a possible influence of peripheral signals to the brain, and potentially the upper layers of the cortex are implicated to that. And I will stop there.
So, thank you very much, Professor. If you please, no questions from the audience because we are a little bit behind the schedule already. Professor, you are very welcome for the next Congress next year here in Cyprus, and we may proceed to the next lecture. Thank you.
So the next lecture is given by Dr. Patrick Pallier. He is coming from Barts in London. He is in charge of projects on neuroprotection and repair after trauma in aging and neurodegenerative diseases. He's going to talk us about how we can treat the blood-brain barrier integrity, because we need to keep it functioning. Thank you.
Thank you very much. And first of all, I want to apologize for being late, but the taxi sent me to the other side of the town, so it was a bit of a nightmare. I'm going to talk to you about the effects of long-term leucine and isoleucine treatment on blood-brain barrier integrity and behavioral and cognitive dysfunction in male biopsy mice. So this is very different from what you've heard already, but this preclinical research is about some of the risk factors for developing dementia. And we do know that these risk factors, many of these risk factors are modifiable. And the ones that are easiest to modify are those that are linked to your lifestyles. And some may be difficult to control, just like smoking or excessive drinking, but this is achievable. Others are easier to attain, like a lack of mental, physical, and social activities, or feeding ourselves an unhealthy diet. And for instance, eating too much processed meat or consuming too much salt. And if the constant, persistent exposure to some of these risk factors are not being addressed, then they can lead to poor health, among which the development of obesity, insulin resistance, type 2 diabetes, or gut dysbiosis. And there is one thing that many of these risk factors have in common, is that the persistent exposure leads to, and this is also seen in Alzheimer's disease, can result into increased circulating levels of the branched-chain amino acids, leucine, isoleucine, and valine. So these are essential amino acids, and they're very important for the brain because they are the primary source of glutamate synthesis. And we know that these amino acids are neurotoxic and the metabolites as well, because this is what happens in maple syrup urine disease. In this disease, there is a defect of the enzyme that is supposed to oxidize the alpha ketoacid derivatives of these amino acids. And the patients have been characterized by very severe neurological and developmental delay and an array of cognitive deficits and behavioral dysfunction, and also presence of markers in the serums of neurodegeneration and myelin disruption. Now, raised levels of these amino acids are also very well known to disrupt the blood-brain barrier. And we do know that blood-brain barrier dysfunction is a pathological feature of Alzheimer's disease that may occur very early on in the course of the disease. And therefore, with my colleague Simon MacArthur, we hypothesized that there may be a pathophysiological link between poor health and lifestyles, raised circulating levels of branched-chain amino acids, blood-brain barrier dysfunctions, and early cognitive deficits and behavioral dysfunction. So Simon wanted to test this in mice, but he didn't have the expertise, and he turned to me to try to set up a behavioral battery of tests to assess very early, very subtle cognitive and behavioral deficits in mice after supplementing them chronically with high doses of either leucine or isoleucine. We started treatment from 9 weeks of age, and after 4 weeks of treatment, I started testing the mice in this battery of tests that I'm going to explain one by one. And at the end, we sacrificed the mice. We took the brain and many tissues and the blood. A lot of work is being carried out on these tissues at the moment. So first of all is the nesting test. Why the nesting test? Because this is a mass equivalent of activities of daily living. And this is something that the mice do very routinely, and that seems to be easy, but actually it does require perseverance, it does require executive function. And executive function impairment is something that signed the start of dementia in Alzheimer's disease. Now, the idea is that you give a piece of folded paper in the cage to the animal, and you come back 23 hours later, and they are supposed to make and build a very nice nest. Now, if you look at the control mice in here, this is not a very nice nest. Actually, it should not look like this. This was the first time we were doing this. Maybe that was the wrong paper. Maybe they didn't have enough time. But a nice nest should be very shredded, and normally the mice pulled it on the corner of the cage, and the height should be higher than the mouse, so that they can be very snuggled into it. You won't see this 3D aspect on these photos, of course, but what you will see is that the treatment had no effect at all on this task in here. And if anything, the leucine, and especially the isoleucine-treated mice, performed better and made beautiful nests exactly how we wanted them to be. So they scored the highest score of 5. We couldn't even run statistics on this graph. But this is not very surprising, because we know that in humans, supplementation with essential amino acids improve cognitive function, and especially executive functions. And you also have to remember that in this situation, the mice are in their own environment, very quiet environment. They do this overnight, nobody is disturbing them, and everything is fine. Now, this is another story in this test. So this is a test that I call usability. I invented it and created it, and this was published years ago. And why usability? Because sleep-wake disturbances are a common feature of neurodegenerative conditions. Now, the idea is to assess the reactivity of the mice to mild disturbance. You take the cage from the holding room, you move it to a quiet room, and then you open the cage. And the experimenter stays still, put in front of the cage, and acts as a distractor. And then, because the mice are very curious, the first thing they do is that they start assessing their environment, and they see you. And then they start exploring and leaving the nest. And what we do is that we take the mouse by the tail, transfer them to a waiting cage, and we can assess the number of mice that are being disturbed by the process over time. And we assess this over 5 minutes. And what you can see is that after treatment with isoleucine, we didn't find any impairment. But this was very different with leucine treatment, and we find out that they were a bit more timid, a bit more reluctant to get out of the nest. And I don't know if this is down to a problem of usability, or if the mice are showing a source of an anxious-like phenotype, because this is typically a condition that is very safe. The mice are very used for the cages to being opened. They should react normally to cage opening. Now, this was done while the mice were very quiet in the holding room. I did this for a couple of days, and then one day, the day after, I arrived in the holding room, and the animal technicians had put the music on, and the tap water was on, the hood was on, there was a lot of noise, and the guys didn't understand what I needed to do, because you needed to have the mice very quiet before you run the test. And this ran for days and days, for 3 days in a row, and I thought, well, after all, I'm here, I'm going to collect, because if you don't collect data, that's lost data. So what happened in the noisy environment was, in fact, very interesting, because in this case, all the mice stayed for longer in the nest. They were very reluctant to get out. They were very stressed. But the isoleucine-treated mice were the ones that were the most affected by this acute stress condition. So I get that they are most susceptible to acute stress. The next test that I use is a test that I invented years ago, and I validated in the mouse model of Huntington's disease, but it was never published. But in this task, I take advantage of the fact that the mice on the rotor rod, they have issues maintaining balance. And then I flank the side of the plates that are running with the rod with cues as a distractor. And this is what I see as an equivalent of the flanker task. A flanker task is being used in the clinic. So, for instance, you need on the red rectangles here, the patient needs to focus, for instance, on the arrows and need to type on the computer which arrow in which direction it is. But it is being flanked by either congruent or incongruent arrows that act as a distractor and then will impair the cognitive function. That's the same idea in here, and I use two types of cues. The first cue, I called it the normal speed cue. This is a normal speed cue because visually, when it turns, it gives to the mice quite a nice visual feedback of the actual speed of walking. And I know that these cues are distracting because normally on rotorod, the mice need to learn. There is an element of motor learning in here, and usually after 3 days of training, they start getting 50, they can say 55 seconds, and then they plateau close to 60 seconds. Here, after 6 days of training, they barely achieve 50 seconds on the road. And then I don't tell the mice at all, and then I change the walking speed, a speed that they never encountered before. And you can see that they drop the performance dramatically. And then I introduce another cue, which I call the low speed cue, because in these cues, in this type of cue, the patterns, the succession of patterns gives a feedback of a speed that is much lower than the actual rotating speed of the road. And in this condition, the mice perform even worse. But the leucine-treated mice, they perform significantly, similarly to the control. However, it was another story with the isoleucine-treated mice. They performed significantly worse when this cue was being introduced. And I suspect that these mice are more easily distractible. This is the size of a normal open field, and normally the open field is based on the fact that the mice ate open arenas that are very harshly lit. I'm using a smaller open field because I wanted to have less aversive conditions with 3 different arenas. The periphery, which is close to the wall, which is being felt safe by the mice, an intermediate zone, and the center zone. And normally this is run in plain light, and you use a very harsh light. And if I wanted this to be very aversive, I would use 600 lux of light. And I use very low light to make sure that this was not felt very aversively by the mice. And in these conditions, the freezing episodes are normally correlated, and they correlate with anxiety-like behaviors in rodents. And you can see that the treated mice displayed more freezing, significantly more freezing episodes, but even very significantly more for the leucine-treated mice. And the leucine-treated mice also spend less time doing unsupported rearings. Unsupported rearings, they are vertical exploration. That's when the mice are trying to find a solution from the center of the open field very often to try and escape. So, and you can see that despite the fact that I try to have less aversive conditions, the mice are avoiding the center a lot. They travel less in the center. So the task is still being felt very aversive. But what I also noticed is that this time I was running statistics, when there was a main treatment effect that was borderline, this was done to the isoleucine treated mice. And these mice explored much less as opposed to the other treated mice. And what I suspect, so less distance travel, more time immobile. And what I suspect is that these mice are different. They are displaying different reaction to the task. And this is speculative at the moment. We'll need to do a lot more work to complete this. But I suspect that the leucine-treated mice display more like a more anxious-like phenotype. But the isoleucine-treated mice may be more susceptible to acute stress. That we've seen this already in the other task.
Professor,
finally,
we have another 2 minutes.
Okay, so I'll go very quickly on the zero-maze. The zero-maze didn't give any significant difference between the mice. However, the isoleucine-treated mice, they seem to show an increased impulsivity. They tend to decrease the latency to the first entry to the open arm. The open arm is the anxiogenic arm. So it doesn't seem that the treatment affected any of the gross locomotor function of the mice. I didn't put the results of the novel object recognition, which is linked to episodic memory. We need to validate this, but it seems to be that there is no deficit in memory, and that the other deficit that we see in anxiety-like behavior, in distractibility, impulsivity, maybe very early disturbances before any deficits in memory happen. And we also find out in these mice that treatment with isoleucine disrupted the blood-brain barrier. And therefore, we propose that chronic treatment with isoleucine has a deleterious effect on the blood-brain barrier and leads to increased reactivity to stress and impaired attentional processes. We don't know if this would lead to increased anxiety, but we know that anxiety often occurs early in the course of AD, and that attention is the first non-memory domain to be effective in AD. So maybe reducing isoleucine intake to slow or postpone the development of dementia symptoms could be one likely strategy. And I want to thank Professor Patricios, of course, and my collaborators, but also the brilliant students from this university who join me most of every summer to do some research with me. Thank you very much.
So, Dr. Paller, thank you very much for the presentation. Unfortunately, we don't have much time because Professor Gravanis? Sorry for that.
Very interesting work. It reminds me, your work, the story of having a peripheral cause for neurodegenerative conditions which can be facilitated entering into the brain through the blood-brain barrier opening. And you know very well the story about Parkinson's disease and alpha-synuclein in the GI tract. So I think that your thinking is out of the box for AD, adding the possibility of having some, deleterious actually signals, and through facilitation of the opening of the they enter into the brain and damage the specific neurons. So would you kindly comment on that?
Well, we are investigating this at the moment. So we've done a lot of in vitro work, and the in vitro work shows that the 3 different amino acids do have different effects on the permeability between cells. Isoleucine increasing the permeability, valine doing nothing, and leucine doing exactly the reverse. So at the moment, we are investigating this a lot because this is quite surprising, and this has been validated by many different kinds of tests. And of course, these are models. We model this. We don't put the 3 amino acids together, which are some in vivo work that we would want to do in future. But at the moment, it seems that these amino acids may have different effects, and some of which may go through increasing inflammation, for instance. But again, this is too early to talk about it because this is the work of my colleague Simon McCarter at the moment, and we're doing a lot to explore this aspect. I was just in charge of the behavioral neuroscience, but we work collaboratively on trying to solve these questions. Thank you so much for your
Well, thank you very much. I think that we are moving to the next session. So I would like to ask the next.
For the next presentation, we invite Dr. Pavlos Kostas and Dr. Violeta Anastasiadou to take their places as chairs. Please.
Ladies and gentlemen, good afternoon. It's an honor to introduce the first speaker of the session, Professor Nikolaos Grigoriadis. Professor Grigoriadis is a distinguished expert from Aristotle University Thessaloniki and a leading clinician at the Heva University Hospital in Thessaloniki, Greece. His work is focused on exploring immune-mediated mechanisms in neurological diseases, particularly the role of complement activation. Today, Professor Grigoriadis will share his insights in a lecture titled Complement and Neurological Disorders,
shedding light on the intricate interplay between complement pathways and neurological health. Please join me to welcome Professor Grigoriadis.
Thank you very much, Mr. Chairman, for your generous introduction. I'm really grateful for being here with you once again, this year, and I'm definitely grateful to the organizers, and particularly to Professor Patricios, a good friend and distinguished scientist and collaborator all these years, for giving me the opportunity to be here and share with you some updates about the complement in neurological disorders. Complement, which is an old player in immunity, though a new target in neurology. And to tell you the truth, while I was a medical student, we all knew that there is no possibility that anything might be asked from us during examinations from the complement. It was all the time in the material we had to learn for our examinations, but we knew nobody was paying any serious attention on it. But after so many years, it came out that complement is a key player in neurology, and these are my disclosures, and this is what I will try to do within the next 20 years. Before I start, I will kindly ask you to allow me, although I haven't gave any notice to the organizers and Professor Patricios, I would like to dedicate this presentation to my beloved Ioannas Karousis. He is the son of our esteemed and well-known colleague and professor, Dimitrios Karousis, from Israel, from Hadassah. His son was assassinated recently by terrorists in Tel Aviv while being in his task for his project by his university. He was an excellent student of the School of Architecture, and you may understand that this was an awful event that had a great impact and still has in our life, of course, primarily to his parents' life, but also to my life and my wife's life, Tina's. We are the godfather and godmother of Ioannas. So in his memory, I would like to dedicate this speech. And finally, because on Sunday we have his memorial ceremony in Greece, in Cyprus, and in Israel. So the complement system contains more than 30 proteins that are present in blood plasma or on the cell surface. And the three major functions this system has is the opsonization of foreign molecules, the increase of inflammatory response via short peptides called anaphylatoxins, and the lysis of the pathogenic microorganisms via the formation of a pore in the lipid membrane. The very last mechanism is also valid for killing not only microorganisms, but also body cells, of course, in the central nervous system. This is the complement system, and we all know that there are three different ways that this system is activated. We pay much attention, of course, to the classical pathway where in order to have the activation of the complement, we need to have autoantibodies. It is the autoantibodies that invite the complement to become activated, and finally, as you can see here, after this cascade of events, it may finally reach to this MAC, the so-called MAC, the membrane attack complex, which is a pore on the cell membrane, and via this pore, the cell becomes degenerated and finally killed. So I will start by the role of complement. I will start from the periphery and then I'll go to the central nervous system. The role of complement in the neuromuscular junction and peripheral nervous system immune-mediated disorders, and I'll start with the more classical neuromuscular junction disease, which is the myasthenia gravis. Here we see three different mechanisms in order to have myasthenia gravis. The first one is that autoantibodies go and block the receptors of acetylcholine receptors in the metasynaptic membrane on the muscle fiber. The second mechanism is that one autoantibody against the acetylcholine receptors may block two neighboring receptors, thus leading to the receptor internalization and finally degradation. And last but not least, it is the complement that it is activated and brings problems in the neuromuscular junction function. But it's not only these sound a little bit more functional, let's say, mechanisms. This one is really very important. Why? Because due to the complement activation, there is a massive destruction, morphological destruction of the neuromuscular junction. So we may understand that this mechanism may lead to more permanent neuromuscular junction malfunction. And whether it is important, the complement or not in this process has been also identified in this wonderful paper made by Angela Vincent in Oxford group, where they found that in anti-acetylcholine receptor antibody negative patients who were exhibiting myasthenia gravis like clinical symptoms, in the biopsies it was found that there is an complement activation already there, despite the fact that no antibodies were able to be detected in the serum of these patients, thus meaning that it's not the antibodies the absolute necessity for the induction of neuromuscular junction. And of course, this has also been further supported by experimental studies, as those two here. You can see that animals that had been deficient to C6, the parameter of the protein C6 of the complement, did not exhibit any clinical disease after myasthenia gravis induction, compared to the wild type, whereas by the time the C6 was substituted in these animals, the animals exhibited very nicely myasthenia gravis clinical signs. And here in this experiment made by Mihailidou and